us about CRISPR? Let's find out. So what is CRISPR? CRISPR is a method of genetic engineering. It stands for Clustered Regularly Interspaced Short Palindrome Repeats. And right now, it's the most efficient and least expensive way to edit genes. It was found in the immune system of bacteria and archaea. And CRISPR itself is basically an archive. Let's say a virus attacks an organism. If the organism survives, then it will store a copy of the virus's code inside a string of DNA called the CRISPR locus. If the virus then returns and attacks again, the organism makes an RNA guide, which uses the code it had previously saved in the CRISPR locus and tries to find a match of that DNA. The guide RNA then tells a protein called Cas9 to cut it out of the organism system. The CRISPR system is incredibly precise and highly programmable and can be used in any kind of cell. Many genetic changes can be caused, including genome editing, epigenome editing, and transcriptome regulation in plants. What are its uses? Well, CRISPR could change not just the niche sphere of scientists studying gene modification, but CRISPR has the potential to change the way all of us live completely. In 2016, CRISPR was successful in removing the HIV strain in 48% of tested rats. Studies show demonstrated eradication of HIV-1 in latently infected human cells as well, meaning CRISPR-Cas9 could potentially defeat viruses like herpes or HIV in the human population. Not just viruses though. Genetic diseases also have the potential to be eradicated because of CRISPR. Most genetic diseases are caused by a single irregular letter in the organism's DNA. With CRISPR, we have the ability to locate and alter that letter, avoiding genetic diseases altogether. We could also use CRISPR to genetically modify our food in beneficial ways, such as making them resistant to extreme weather, drastically reducing the severe impact that climate change has on humans. In 2019, CRISPR has been shown to be able to accelerate both breeding and germplasm development in plants. But as of right now, it's mostly just used by scientists for research, to learn more about genes and speed up experiments. Are there alternatives to CRISPR? The answer to that is yes, but they're not very good. CRISPR itself was discovered fairly recently, though the year could be debated depending on your definition of discovery, but the discovery of the use of CRISPR with Cas9 is commonly attributed to Jennifer Doudna, Emmanuel Compertier, and Feng Zhang in 2012. But to be clear, genetic engineering had existed way before this. It was actually discovered in the 1970s and has become integral to our everyday life. For example, GMO foods. GMO foods are foods that are genetically modified, and most often they're able to withstand herbicides or appear fresher for longer. Before CRISPR, scientists had three main methods of genetic engineering. Transfection, a solution that enables the edited DNA into lab-grown cells. But this typically only lasted for a few days, and this behavior was often irregular and hard to control. There's also modified viruses, in which an edited virus can deliver the gene. The viruses then integrate the genetic materials that they're carrying with the host's DNA. But for this, there's nowhere to control where the virus puts the DNA, and it can lead to unwanted effects if it gets placed in the wrong spot. And the third way is nucleus proteins. These proteins are able to cut sequences of DNA, but in order to do so, the scientific engineer, they have to create an entirely new protein structure for each edit each time. And this takes copious amounts of money and time. The common thread is that all of these alternatives are difficult to control, often unpredictable, and take a lot of time and money to get done. As I mentioned before, CRISPR uses RNA to locate where to cut. RNA is easier to make than protein structures. In total, it's faster, cheaper, and more accurate. Though CRISPR is the most reliable method of genetic engineering, it's not perfect. Sometimes the guide RNA makes mistakes and misidentifies strings of code. This is known as off-target, and this has about a 50% chance of occurrence. And while that is more accurate than most of the other methods, it's still not ideal. Another downside is that CRISPR and gene editing in general forces us to acknowledge moral questions about this kind of technology. So what could this mean for humanity? As of right now, CRISPR is mostly just used in research for scientists to learn more about genes, fast-track experiments, or speed up other research. But once we learn the full capabilities of CRISPR, the genetic engineering possibilities are vast. If we're thinking big, this could potentially alter our society irrevocably and eradicate genetic diseases. But once we begin modifying humans, where do we draw the line between health purposes and vanity traits? If we're immune to Alzheimer's, why not have perfect vision, strong muscles, or enhanced intelligence? This brings up the question of designer babies, in which the genetic attributes can be predetermined by the parents. This brings up moral and ethical qualms, as well as the question we just don't know the answer to about what happens when altered genes enter the genetic pool when passed down by generation. We can't predict possible outcomes. This opens up a big debate. But all in all, that possibility is still far off in the future. As of right now, CRISPR has a positive effect on humanity and helps the realm of scientific research.